Hello, everybody. I have been asked by some of you whether it would be possible to provide the presentation we had at the International Congress of Entomology in Japan in August. Actually, it's very fine to do. I was just too busy during the semester, so now finally I'm a little bit free. I can try to record. I was naturally responsible for the hydrophiloid part only. The hysteroid part was done by Tomáš Lackner, who is not here with me now. So I will try to reproduce his part somehow. But naturally, the original version at the Congress was surely much better. In the original version, we also had some strict time limit. Now I'm not that limited, so I will try to be a little bit slower because some people complain that we click through the slides a little bit too quickly. Cool. So I will remove my face. That's not necessary for this. The presentation was part of the Beetle Systematics and Evolution Symposium, which was dedicated to John Lawrence. And we were basically asked to provide some update on what has been done in phylogeny, evolution, and systematics of these two groups. So this is the topic of the lecture. Just to navigate through the beetle tree of life, so these two lineages are part of Staphyliniformia, which you can find here. Here are the groups which we will be talking about, and they are basically consisting of two superfamilies, Histeroidea and Hydrophiloidea. Both are supported as were supported as sister groups by all means of evidence, by morphology, by single genes, by genomics. Sometimes some groups, especially John Lawrence and around, considered these two groups as one superfamily, Hydrophiloidea, which is naturally possible. Whether it should be one or two, it's more or less formal thing. It's nothing scientific. And here we follow the more traditional view of two superfamilies, which is mainly kind of maintained because the people working on histeroids are rarely touching hydrophiloids, and people working hydrophiloids are rarely touching histeroids. There's some kind of isolation between the workers. So that's why it is so far kept separately. I think is the main reason. Would be actually nice if we would start to study a little bit together, to compare a little bit more, which is also why I was very happy that we were kind of asked to combine both groups into one presentation. Both groups have something in similar and they differ in some aspects. So the similar thing is that both superfamilies, have some small basal or early branching families. So these are Synteliids and Spheritids in Histeroidea, both with seven extinct species described at the moment. In Hydrophiloidea, there are five smaller families. You can see that except of Sparcades, which have only 18 species, the others have a little bit more. They are a little bit more species diverse than the hysteroid small families, but still they don't really reach 300 species. In contrast to these tiny species poor lineages, there are two big kind of lineages with thousands of species. In Histeroidea, it's Histeroidea naturally. In Hydrophiloidea, it's Hydrophilidae naturally. And within these lineages, there was a huge radiation of morphotypes, lifestyles. They are widespread and so on. So actually, the main diversity of both groups is within these two families. So whenever you probably meet some hysteroid or hydrophiloid, it's most probable that what we have in front of you is representative of one of these big family. What is similar among them is also the fossil record. So it dates back to late Jurassic in hydrophiloids and to the Cretaceous in hysteroids. You can see that not all families are actually present in fossil record. We have quite many fossils of hysteroids. There's one fossil of Synteliids, but no Spheritidae. In Hydrophiloidea, we have fossils for all families except of the small Georisidae and Epimetopidae. A special thing in Histeroidea is the extinct family Cretohysteridae. which was published and described in this very nice paper in Systematic Entomology. It is extinct. It has one genus, one species from Chinese Cretaceous. In Hydrophiloidea, we don't have any extinct family level lineage yet. As it concerns the biology, both superfamilies differ quite a bit. So Histeroidea are terrestrial species. You find them in any kind of terrestrial habitats. And adults and larvae basically share similar lifestyle, a similar habitat, and they are both predatory. There are some exceptions in hysterids, which may be liquid feeding, but generally the adults or larvae share the same kind of lifestyle, so predators. This is in contrast with Hydrophiloidea, where the adults are saprophagous. They feed on some organic remains, detritus, and so on. But larvae of all groups are predatory. In Spurcates, it's a little bit special. The larvae are filtrators. They filter the organic, like pieces of organic remains and from the surface of water. The family relationships have been unclear for a very long time. When we try different data sets, different methods, we always get some different topology. So if you actually use larval morphology, adult morphology, different genes, genomics, it provided very different results. So we actually recently submitted a paper where we tried to generate a huge genomic data for all the families for the first time, more than 4,000 protein coding genes. This actually helped a bit for Hydrophiloidea. We got very stable topology, which is the one shown in both these trees. So you can see that they actually have kind of two main lineages. One consists of Georisidae, Holophoridae, and Hydrochidae. The other of Epimetopidae, Spurcaceae, and Hydrophilidae. In Histeroidea, unfortunately, we did not get any stable topology because the position of syntellids it's not really clear. What we can definitely reject is that syntellids are definitely not sister group of hysterids. This was not supported by the data, but we are not able, based on this data, to distinguish whether syntellids are sister to spheritids or sister to all the other hysteroid families. So these are also these two topologies show you these two trees show you the two topologies which both can be correct. We did lots of topology tests, and it seems that this problem, this instability, it's not based on the kind of methodological issues. It probably reflects some biological problem behind, and one of them can be the ancient quick radiation of hysteroid families. Okay, so now let's go a little bit into each of the superfamilies. So for hydrophiloids, what has been done before I actually started to work on this group, was lots of work by Michael Hansen, who actually did the first phylogenetic analysis based on morphology. Based on then, he adapted the classification. He also did the revision of genera worldwide, and he made a word catalog of all species. So it was actually huge work on which we are still building. And roughly at the same time, Miguel Archangelski, the Argentinian researcher who was studying in Ohio, started to work on larvae. He published a very nice book on American larvae. And all this together actually forms, until now, a very solid base for the further research. So everything what I will show you later is actually would not be possible without these two people and their really amazing work. Okay, so what do we know about the small families? The first interesting thing which we actually discovered by this genomic phylogeny is that these two small families, Georisidae and Epimetopidae, are not closely related. They are quite similar in lifestyle and quite similar on how they look like. So they were quite frequently considered as closely related, but they're actually not. As you can see, they are always the basal or early splitting lineage of of the two main clades of Hydrophiloidea. And the interesting thing that they're actually not aquatic, that live in the riparian habitats. These are kind of wet sand or gravel at sides of the streams or rivers. So they don't go underwater. They basically dig in this wet sand. And similar thing, not digging, but not living in water, but on this river banks and river sides, or kind of sides of the water bodies rather than inside of water, is also present in the larvae of halophorids. So in halophorids, the adults are normally aquatic. You can find them normally inside of water as all the other water beetles, but the larvae are actually not in water. They are at sides of the water bodies. So based on that, we actually can propose a new hypothesis on how the evolution of this group worked. It is quite probable, based on the ancestral analysis we made, that the ancestor of hydrophiloids was actually a riparian species. That would explain many things. For example, it would explain why we cannot find older fossils, because naturally the riparian habitats are not very suitable for fossilization. It would also expect that they colonized aquatic habitats twice. So once in sparcates and hydrophilids, once it's in hydrochaetes and halophorids. So it actually somehow puts everything we were thinking about or considering in this group upside down, because until now we were actually not in doubt that the aquatic ancestral hypothesis is true. Now our data kind of a little bit challenge that. So we will need to do a little bit more studies to figure out which one is more probable, but definitely both alternatives are now have now to be considered. As it concerns the systematics of the small families, so we have actually two families, halophorids and epimetophids, in which lots have been done, mainly thanks to Robert Angus in halophorids and Phil Perkins in epimetophids. So most of the species are surely described. Time to time, naturally, there are some new discoveries, some single new species described, but we don't expect kind of many new species to be discovered. DNA work would be cool now, so all the systematics was done based only on morphology, so it would be cool to start to work on DNA and try to figure out whether this supports the view. But otherwise, these two things are done. For sparcates, there's a revision by Franz Hebauer from 1997, I guess, but it is kind of not very careful, and it would actually need a modern study to kind of make things a little bit more clear, and also the DNA would be cool. And then we have two lineages, hydrochaetes and georhisids, for which we know that actually they have many undescribed species. They were totally understudied. Phil Perkins actually did in last years many studies of American hydrochaetes, which and he described many new species, but actually the same situation with really unexplored fauna is definitely the case of Africa and Asia and also Australia. In Australia, it was revised, but there are still some new species expected. And for georhisids, it was actually never touched properly. There were some studies in 1950s, 60s, but since then nothing was done, and there are many undescribed species present in every piece of the world. For Hydrophilidae, the largest family, we actually have quite a good idea about the internal kind of topology or relationship among the main lineages. This was kind of first done in our study with Andrew Short, which was based on six gene molecular phylogeny. Based on that, we somehow rearranged and adapted the classification of the group. And surprisingly, all subsequent DNA studies and morphology studies of larvae and adults so far seem to support this view. So hopefully or likely we are quite close to reality. So what we need to, what is interesting to or remember or realize is that hydrophilids actually include aquatic as well as terrestrial lineages. And you can see here that the aquatic ones, which are marked by the blue text, are the kind of early splitting or basal lineages, and there was actually one major shift to land somewhere during the Jurassic. So all the terrestrial groups are actually related, and they have a one common ancestor. So what is the main effort now? We are now trying to actually sample most genera and try to figure out their relationship, and also sample more and more species to try to delimit the generic to delimit the genera more clearly. And actually also would be good to get as much information as possible on the larvae and biology. For the genus level kind of revisions and systematics and so on, there are some differences between different lineages. So as you can see, we have some lineages like Acidocerines, Hydrobiosines, and Hydrophilines, which are done and published. Then there are some lineages which are actually done but not published yet, or nearly done or in progress and not published yet. And few lineages, Amphiopines, Parasimus group, and Spiridians were not even started. So still lots of work to do, but actually we are in process to become closer to reality and to understand the evolution of this lineage of hydrophilids much better. Just a nice examples of the big progress which has been done. The best example in recent years is Acidocerines. It's a big subfamily of primarily aquatic hydrophilids. Andrew and Jennifer did lots of work on this group. They published the phylogenetic paper in Systematic Entomology, and then generic revision in Zookeys, and kind of redefined the genera, re-establish the classification. Now it makes actually much more sense, and Jennifer did very nice comparative morphology of the genera. They also discovered very cool things, for example, that Guiana Shield in South America hosts totally unexpected diversity and previously totally overlooked, which actually consists both on very old lineages of Jurassic age and the quite young Cenozoic radiations. So it's actually a very cool area for this group. And they naturally also describe many new species. We were also trying or starting to do something similar in the terrestrial groups. The main effort now is in the most crazy lineage, which is called Megasternines, for which we did the molecular phylogeny based on the sampling which was available at the moment. And now we are trying to redefine the genera, put more species in, and slowly revise the group step by step. It's actually lots of work, but fortunately Zuchi joined us, and he is really hard working on the Asian groups now. The other terrestrial groups are in progress. They are not yet published, but we have actually lots of data, and hopefully we will publish at least some of the results quite soon. For larvae, you can see that all is green. So we actually have very good knowledge about the larval morphology in hydrophilids. In some lineages, we even have data for all genera. This is mainly thanks to the really massive work on larval morphology by Yusuke Minoshima in Japan and the Argentinian team, Patricia, Georgina, and Miguel Archangelski. So for that aspect, we are actually really very lucky that we have lots of information. Naturally, it also goes together with some data about biology, so we actually have quite a good idea of how most of the things are living, even though more studies would be necessary. For fossil data, so actually you can see that we don't have fossils for most lineages of hydrophilids. And actually, only 3 lineages are so far represented in fossil record of the Mesozoic. So we have some Cretaceous fossils, and there's few more Cenozoic, but actually many lineages are absent. And specifically, the older terrestrial lineages are so far absent from fossil record. This actually a little bit complicates the dating. So if we want to do the molecular dating, we don't actually have that many fossils to calibrate the tree. And another thing which is kind of limiting us a lot is that we still don't have any good morphology matrix for adults. So once there are, naturally most fossils are adult beetles. So once we have new fossil and some kind of unusual one, it is not that easy to place it, and we are not able yet to do any formal family-wide analysis because the data are simply not present. That would be also cool to sit down and compile the morphology data for adults. For larvae, there's quite good data compiled by Miguel Archangelski, so for larvae it's actually quite easy. Naturally, because we have all this basic knowledge about the relationship among the main lineages or among genera, the timing of their splits and so on, we are also able to slowly start to focus on some kind of interesting evolutionary or biological questions. For example, we try to analyze the biogeography of many groups. Here's the example of the megasternines, where we kind of suppose that there were many intercontinental shifts during the Mesozoic. Also, there are very many good studies on biology and behavior, mainly by the Japanese colleagues. We are also now working on the study on the swimming behavior and evolution of that. We naturally started some molecular things. So there's a paper published on karyotypes, and we are working on the paper on genome sizes. And we are now actually also completing the first completely annotated genomes based on the kind of model taxon, the Vedrok Agramhydrus from Taiwan and Iriomote. So hopefully soon we will have much more data, and we will be able to actually address more than just systematics and species diversity, but also some clearly evolutionary topics. Cool. So that's it for hydrophiloids. For histeroids, there are also lots of work done. The older kind of founding research for histeroids to establish the classification and so on was done mainly by Axel Reichard and Oleg Krzyżanowski. Both of them were based in Russia, or the area of Russia. Then Slavomir Mazur, the Polish researcher, did lots of studies on the kind of species level systematics, and he also compiled the catalog, the catalog of all species. And Professor O'Hara did lots of work on kind of establishing the classification as well. So this is actually what we have now. Now we can go to the individual families. For the small families, we have Syntellids, which have now 7 described species living today, plus there are 2 species to be described. They have a disjunct distribution, so 5 of them are living in East Asia, 2 are living in Mexico, and those which should be described are also living in Mexico and Guatemala. There are species living in temperate forests in Asia. In Mexico and Guatemala, they are actually in the pine forests. And there's one species, the Centellia westwoodi, which actually lives in arid areas. It is specifically living in the rotten columnar cacti. In general, you can find them in rotting logs. And they are both, the larvae and adults are predatory, so they are evidently hunting some insects in these habitats. There's a single fossil known from Barney's amber, and the phylogenetic relationship, unfortunately, among the species are not yet studied because they're actually not that easy to get. Larva is only described for one species, for Centellia hysteroides. Okay, second small family is Spheritids. It has, I think, 7 described species now. This may be a mistake. They are holarctic, so they are also confined to the northern hemisphere. 4 of them live in China, 1 of them lives in kind of across Palearctic, and 1 species lives in North Africa, I think. They are mostly living in temperate forests, or some of the Chinese species also reach the alpine areas. There are no fossils. Larval morphology is actually very poorly known. There's only the first install larva of Spheroides glabratus described by Nikitsky, but the description is quite poor and it would need much more study. And in generally, these are kind of rare beetles which are poorly known and rarely collected. Thomas actually collected many of Spheroides glabratus in the boar cadaver pitfall traps in Bavarian Forest Park in his recent fieldwork, but no larvae were found. So actually, this is probably not the normal habitat for development of this species. So lots of the research needs to be done about this nice small group. For historyds, for the largest family, so there was naturally they have very many species, as I mentioned, 4,800 roughly. So the classification was kind of historically established, but then actually challenged a little bit by the molecular data. But unfortunately, still the molecular data are very limited. There are several studies. So Mike Caterino used 18S, Duane McKenna 28S and CAT. And then the Chinese colleagues used 4 genes and morphology. So they somehow got some results which actually challenged the kind of traditional classification. But as you can see, even the species sampling is still quite poor, right? So we have something like 50 plus species only included in the analysis, only few genes. So there's still lots of work to do to actually understand the phylogenetic relationships of historids and to kind of propose their classification. But still, this data help to solve some part of the confusions. For example, the two kind of weird-looking subfamilies, the Tripeticines and Tripeniines, were consistently recognized as internal clades of Abrainae, and they're already now actually moved there, and these subfamilies are kind of synonymized. This actually corresponded to larval morphology, so it was not really unexpected, but it was actually cool that DNA confirmed what Al Newton was thinking about for a long time. The only group which is kind of studied a little bit more, thanks to Tomáš activities, is Sapriniines, so one of the subfamily. Tomáš accumulated lots of species and genera across, so we actually have quite good idea about good sampling and basic idea about how they are related to each other. Still, the taxon sampling is cool, but the problem is that they still only use 2 genes, so it can be still a little bit preliminary. And it looks like, at least based on the data which we have now, that the diversification in Sapriniines actually somehow corresponded with the mammal diversification. For larvae, not that much is known as in hydrophilids, so we have data about 10% of history genera only, kind of known or described. Unfortunately, many of those are not published. And if you want to study the history of larvae, actually you have to go to Field Museum to see Al Newton's collection, and to Ohio to see Pete's Kovarik collection. They have lots of material, lots of data, lots of knowledge about the larvae, but actually not much is published officially. For fossils, as I already mentioned, historids actually have very rich fossil record compared to the hydrophiloids. So there are also many fossils from the Cretaceous, including many of them in the amber, in Cretaceous amber, and there are some younger fossils too. Many of these Cretaceous fossils represent extinct genera, and many of them combine some morphological structure which are absent from modern lineages. What is also clear that some lineages which are relatively species poor or not that diverse today, like Ontophyllines, Heterines, and so on, were much more diverse during the Cretaceous. There's lots of work done on the fossils. The most active guy now is Jan Pražák from Czech Republic, who is publishing kind of one paper after another with many cool fossils. For the kind of more advanced evolutionary studies and so on, it's so far difficult to do because we still don't have enough data about phylogeny. So we need much better idea about the phylogeny and classification first. So it was only possible to do some of these first attempts in the Saprinians, for which the data are available. So for Saprinians, which Tomáš did, it looks like that they originated in the northern temperate zone and colonized the southern hemisphere several times. And also it's kind of supposed, based on the ancestral state reconstructions, that the ancestral habitat for the kind of ancestor of Saprinians were the nests of mammals and birds. Naturally, we need to do much more. We need to do behavior, evolution of body shapes, evolution of morphological structure. But for all that, we would need first the much better idea about phylogenetics. So for what to do for the future, there actually still what is really well done and progressing well is the taxonomy. So there are actually many large revisions already published or kind of in preparation. And there are many very active workers worldwide working on taxonomy and systematics of these lineages. So in this, historians are actually very cool group. There are also some monographs for larger regions like Morocco, Corsica, Australia, Levant, and so on. But we still need more naturally. So it's not only about to do some better idea about phylogeny, but naturally we still have many areas of the world where histories were not really studied properly. So we need mainly more and more young people who will actually join and collect and describe species, study morphology, study molecular evolution, and accumulate much more data to actually be able to understand the evolutionary history and biodiversity of this group. We also need much more densely sampling genetic or genomic studies. So that basically means new fresh material in alcohol. So collecting and fieldwork is also badly needed. Cool. Hopefully it was informative, and thank you for your attention. If you have some questions, you can contact me or Tomáš considering the hydrophilids or historids.